Today I am going to discuss with you all about the genetic code which is given in your syllabus in molecular biology. So let's get started now. So about the genetic code, what is genetic code actually? It is saying that genetic code are actually they are set of rules. So genetic code is generally a set of rules used by the living cells in order to translate the information which is encoded within the genetic material into proteins. So the information which is stored in the DNA or in the mRNA, that information is uh, encoded into proteins. So these genetic code are some set of rules that is used by the cells in order to translate the information into proteins from the DNA or mRNA. Okay. Now, whether we know that the translation is the main process for conversion of these genetic material into proteins, and the whole translation process is accomplished by the ribosome, which links the amino acid in an order that is specified by the mRNA. So normally the whole translation process is accomplished by ribosome. Ribosome is the site where translation process occur, and during this translation process what does ribosome do? It links the amino acid in an order which is specified by the mRNA, using the tRNA molecules to carry that amino acid and to read the mRNA 3 nucleotides at a time. So that it simply means that, as given in the figure, tRNA main purpose. So this is your tRNA, right? So this is your tRNA which is carrying a specific amino acid. Here it is carrying leucine, and tRNA have anticodon sequence. You can see right here there is a anticodon sequence. With the help of anticodon sequence, it base pair with the codons in the mRNA. It base pairs with the codons in the mRNA. What are codons? Codons are 3 nucleotide long decoding element. Codons are 3 nucleotide long decoding element that codes for a particular amino acid. So when it identifies this codon, okay, specific for a particular amino acid, like here GUC, this codon codes for leucine. So immediately the ribosome recruits the tRNA which is carrying the leucine amino acid residue. Okay, so that's why they have said that in this lecture they have said what? They have said that this whole translation process is accomplished by the ribosome, which links the amino acids in such a way, in such an order which is specified by the mRNA. That order of amino acid is related with the mRNA sequence, and they use the tRNA molecules, also called transfer RNA. This tRNA is carrying that specific amino acid, and they read the mRNA 3 nucleotides at a time. That means they are reading that codon sequence. They are reading that codon sequence found within the mRNA, and tRNA is then recruiting, the ribosomes is then recruiting that particular tRNA carrying that specific amino acid for that particular codon. Okay, Then it is saying that mRNA specifies the order of amino acids that will further join by peptide bonds. So the mRNA which is present, which is going to be translated, which is using as a template here for translation, this mRNA specify a correct order of amino acids. So it has a correct order of amino acid sequences. It has actually nucleotide sequences, and that nucleotide sequences represent codons. Three nucleotides make one codon, which are located right next to each other, that codes for particular amino acids. So in this way, mRNA is specifying that order of amino acid which will be joined by the peptide bond as catalyzed by the 23S rRNA subunit of ribosomes. Such ribosomes are mainly responsible for the formation of the peptide bonds between the amino acids. And if I say more specifically, the large ribosomal subunit have 23S rRNA in case of prokaryotic organism, which is actually catalyzing the joining of amino acids by peptide bond. mRNA also uses the tRNA molecules which will carry the amino acids and will read the mRNA 3 nucleotides at a time. So the mRNA, they use the tRNA molecules, okay, and the tRNA molecules is carrying that amino acid, specific amino acid. This tRNA will read that mRNA sequences in a manner, in 3 nucleotide manner. So it will read the mRNA sequence in 3 nucleotides at a time, which is called as codon, that is known as codon. And tRNA have an anticodon. With the help of codon anticodon base pairing, it recruits tRNA, allows that particular amino acid to be incorporated to the growing polypeptide chain. Now what is codon? It is written here. Codon is a sequence of 3 DNA or RNA nucleotides that correspond with a specific amino acid or stop signal during protein synthesis. So what is codon? Codon is a sequence. Each codon represents 3 DNA or 3 RNA nucleotides, and these each codon correspond with a specific amino acid or stop codon. Okay, stop codon means it will not code for any amino acid, rather than it will makes a signal so that the translation will be stop in response to this stop codon. Now, so codons are 3 nucleotide long units found in DNA and RNA, and each codon codes for a particular protein, or it may code for the stop signal. Now which codons are the stop signal, I will show you. Now these codons do not overlap each other, so they are not like overlapping sequence, and they are located right next to each other. They are adjacent to each other. So the codons are adjacent to each other. They are right next to each other. Now there is a term called open reading frame. There is a term called open reading frame. What is open reading frame? Open reading frame is the series of codons, okay, that begins with a start codon and ends with a stop codon. So open reading frame, this open reading frame also called as ORF, it is a series of codon, or you can say it is a stretch of codon that begins with a start codon and ends with a stop codon, right? Now open reading frame is the part of the reading frame that has the ability to be translated. So it is that part of that our gene which will be translated to form a polypeptide chain, right? Now the genetic code that I am going to show you, okay, a table has been given. The genetic code that is given here, it is highly similar among all the organisms and can be expressed in a simple table having 64 entries. So let us see that table. Okay, okay. So how to read that genetic code I will also show you. Let us see. So this is that genetic code. Okay, so I am going downwards to show you the genetic code. Yes, this is that genetic code. Well, you can see here in this genetic code, you can see here there is there are different way that there is different boxes, many types of boxes are there, and total number of codons are 64 here. 64 codons are there, and these 64 codons are coding for the 20 amino acids. I think there is 23rd amino acid nowadays it is observed. So it is coding for the 20 amino acids. Okay, 20 amino acids. Now we have 23rd amino acid, but it is saying that 64 codons coding for 20 amino acids. So how to read this codon? Let us see. Look at this. It is written here first position. Here it is written second position, and here it is written third position. So what does it mean? Okay, what does it mean? Let us see. The first position is the first letter of the codon. Second position is the second letter of the codon, and third position is the third letter of the codon. Let us see by zooming into a particular box. Okay, I am now observing this top box. This top box. Let us see. The first letter is U, second letter is U, and the third letter right here is U. So what does U means? Well, this is your U right here. This is your U. First letter, second letter, third letter. So U, this codon represents PHE. It represents PHE. I think PHE is your phenylalanine, right? Just search it. What does the 3-letter code PHE means? I think it is phenylalanine. Then let us see first letter U, second letter U, third letter C. So what does UUC represents? What does UUC represents? Well, UUC represents again phenylalanine. Okay. Okay, let us see the third one. UUA. UUA. So let us see what does UUA means. Here it is UUA. UUA encodes leucine and another amino acid. Then another amino acid, let us see UUG. So the first codon is U, second codon is again U, and the third letter of that codon is G. So UUG represents again leucine. Okay, let us skip. Let us move on to another sections. Okay, let us move to the another sections for you to easy to understand. Let us move to downward. Okay, let us move to downward. Okay, so let us see this one. Okay, so I have to small it down. Okay, so let us see this one, how CAU get its name. Well, the first letter is C. In this entire box, C has been written. So C, the first code is C, second letter is A of this codon, and the third letter is U. So CAU. CAU codon codes for histidine, which is written in 3-letter code as HIS, HIS. Then let us see this one. Let us see this, how it gets its CCC name. The first letter code is C here. As you can see here, I am giving with a different color. This is the first letter C we get, second letter C here, and the third letter C we get from here. So CCC. CCC code for proline amino acid. In this way, the 3-letter codons are being named. So there are 64 codons, and each of these codon code for a amino acid, right? One codon will not code for more than one amino acid, but there are multiple codons which code for one amino acid. Like here you can see UUU and UUC. These are the two codons which code for phenylalanine. Then you can see the best example here. 4 codons UCU, UCC, UCA and UCG. These all 4 codons code for serine amino acid. Okay, then again you see from here also you see CCU, CCC, CCA, CCG. These 3 codons, these 4 codons code for proline. These 4 codons code for proline. It codes for proline amino acid. Well, you can also see some stop codons as well. Stop codons do not code for any amino acid, but they trigger the translation process to terminate. Like here you can see UAA, UAG and UGA. These are the stop codons. So these are all stop codons that you can see here. Okay, now you can see where how the UAA get its name. Here is U, you get the U, the first letter is U here. Okay, second letter code A and the third letter code A. In this way we get UAA. It is a stop codon. So if ribosome encounters this stop codon within the mRNA, it will not recruit any tRNA to come and bind, but rather it will terminate its translation process. Okay, UAG is also a stop codon and UGA is also a stop codon. So in this way you have to study all these codons. Okay, you have to recite them one by one. Okay, so as in the genetic code, as we can see that the code defines that how codons specify which amino acid, right, that will be added to the next during protein synthesis. So the mRNA contain, in this way it contains series of nucleotides which represent codons. 3 nucleotides make one codon, which are located right next to each other, and each code, this genetic code that we can see, it defines that how codons will specify what type of amino acid has to be added next during the protein synthesis. Okay, now there are some exceptions also. A 3 nucleotides codon in a nucleic acid sequence specifies a single amino acid. There are some exceptions like stop codon do not code for any amino acid, but each codon specifies a single amino acid. While the genetic code determines a protein amino acid sequence, there are also other genomic regions that determine when and where these proteins are produced according to various gene regulatory codes. So there must be some gene regulatory codes, and that gene regulatory codes will be read by the regulatory proteins which will determine when and where these proteins has to be produced, right? So there must be some regulatory regions of this open reading frame of this translation process which will determine when and where these proteins are to be produced. Okay, now let us see about the main part about the features of genetic code. Well, in small words, I have said some about the points of the features of the genetic code. First, let us see elaborately about these features of genetic code. The first feature, triplet nature. That means genetic code are found in three nucleotide. That means three nucleotide make one codon. George Gamow in 1954 postulated that each codon is a triplet code. So George Gamow have proposed that each codon is a triplet code. Triplet code means each codon is made up of three nitrogenous base, or you can say each codon is made up of three nucleotides, and each codon is encoding the 20 standard each codon is coding a specific amino acid, and there are about 64 codons. These 64 codons are coding for 20 amino acid. Okay, and that amino acids are the building blocks of proteins used by the living cells. Well, other scientists have also debated that there are four types of nitrogenous base found in our DNA, and upon transcription, the bases in the mRNA are A, G, U, and C. So there is no thymine in mRNA, there is uracil in place of thymine. And this transcribed mRNA will code for the proteins. So four types of nitrogenous base in our DNA, and during transcription the base in the mRNA are little bit changed, like instead of T there will be U, and these transcribed mRNA have that codons which will code for proteins. Now these four bases containing nucleotides in various combination code for the 20 amino acid. So the four bases that is found within the four different types of nitrogenous bases found in the mRNA, at various combinations these bases will try to encode for the 20 amino acids. Now if we try to consider the genetic code to be a singlet, that means one nucleotide coding for a amino acid. Suppose we other scientists have also proposed that if one nucleotide code for a particular amino acid, then what will happen? So if one nucleotide code for a particular amino acid, then it would be impossible, because there are only four bases that cannot code for 20 amino acids. So suppose some scientists have said that there are four nucleotides, okay, and these four nucleotides will code for the amino acids. Good, but there are 20 amino acids. How will these four nucleotide coding for 20 amino acids? If we say that the genetic code is a singlet, if we say that the genetic code is your singlet, that means one nucleotide coding for one amino acid. That is impossible, because there are only four bases and there are 20 amino acids. So if a codon is single code, if a codon is a single code, that is it is made up of one nucleotide, it can then code for only four amino acid, A, C, G and U. Okay. Now some scientists have proposed that the codon must be a doublet code. That means a codon consists of two nucleotides. So two nucleotides it may contain. So if a codon consists of two nucleotide, or if the genetic code is a two nucleotide, then an amino acid, that is an amino acid is coded by two nitrogenous base, then obviously there will be more combinations of nucleotide to make amino acids, but it can form only 16 amino acid if we arrange right next to each other. Let us see that arrangement that I have drawn here. So these are the combinations of doublet code, two nucleotides that makes one codon. Some scientists have proposed actually, but only 16 combinations are possible, and these 16 combinations are producing 16 amino acid, but there are 20 amino acids. Still it is not sufficient for the to code for the 20 amino acid, so the genetic code cannot be of two letters. Now if the genetic code is a triplet code, if the genetic code is a triplet code, that is if an amino acid coded by three nitrogenous bases, then it can form 64 codons, the table that I have shown it to you. As you can see, the triplet code, or if a code consists of three nucleotides, then 64 different combinations of nucleotides can be prepared, which will code for 20 amino acids. Obviously there will be more than one amino acid which will code for a specific amino acid. So there will be more than one codon coding for specific amino acid. So the possibility is in some amino acids may be coded by more than one triplet code. Okay, I think you have understood what I have said. So therefore genetic code is always triplet. The codon is always triplet. Okay. Now, an important part has been mentioned here. An important part has been mentioned here related to the translation process mainly. The genetic message coded in the mRNA molecule. The genetic message coded in the mRNA molecule will be translated into the amino acid sequence of a polypeptide. So the genetic message coded in the mRNA molecule, that genetic message will be translated into amino acid sequence of a polypeptide. And during this process, there will be sets of 3 mRNA nucleotides, which is called as codons. So the set of or sets of 3 mRNA nucleotides are read successfully from 5 prime to 3 prime direction, starting from a start codon or initiator codon, which is AUG. Well, AUG is your start codon. I have not shown it to you. Well, I have shown the stop codons UAG, UGA and UAA. AUG is your start codon, which codes for an amino acid called methionine. Methionine in case of eukaryotes. In case of prokaryotes, it codes for N-formyl methionine. So it is saying that during your translation process, there are sets of 3 mRNA nucleotides that will be reading by the ribosomes successfully from 5 prime to 3 prime direction. Starting from your start codon, the ribosome starts to uh, read the codon sequence and recruiting that particular tRNA that carries specific amino acids for that particular codon. Okay, so I have said AUG codes for methionine, which is a start codon, and it will move on it will go continues to move on towards 3 prime direction until a termination codon, or you can say until a stop codon arrives at the site on the ribosome. So ribosome started to read the codon sequence one at a time from the start codon, and it is moving by reading the codons one at a time towards the 3 prime end until it encounters a stop codon, which will terminate the translation process. Now, since a termination codon does not code for any amino acid, the polypeptide chain synthesis will be stopped. Okay. So the whole process of translation is this actually. Okay. So the whole sequence of mRNA starting from the start codon up to the termination stop codon is called as open reading frame. And ribosome starts to observe those codons one at a time towards the in a 5 prime to 3 prime direction until it reaches to the stop codon, which will terminate the translation process. Okay. Okay. So one thing that I want to say about this reading frame is that suppose somehow a nucleotide sequence is incorporated within this coding region, then it will code for a different amino acid, or it will shift the open reading frame. So it is saying here, since the reading frame is a continuous sequence of nucleotides, addition or deletion of a single nucleotide will result in the change of the codon sequence from that point onwards towards your downstream. So this open reading frame is very specific for a particular polypeptide synthesis. Now suppose in this open reading frame, which consists of these codons, if one of the nucleotides of this codon gets added or deleted away, it will change that codon sequence from that point towards downstream. From that point, the whole codons will get changed and will code for different amino acid. Such type of event is a type of mutation. Well, mutation means where we are changing the nucleotide sequence of the DNA that will codes for an abnormal protein product. That is known as frame shift mutation. Well, frame shift mutation means the open reading frame is shifted due to the addition or deletion of a nucleotide sequence in the coding region. Let us see in the figure for you to easy to understand. Well, you can see a series of codons has been written here: AUG, CGA, UUA, UAC, GGG. And each of these codon is coding for a particular amino acid. Like AUG code for methionine, it is a start codon. Then CGA code for arginine, UUA code for leucine, UAC code for tyrosine, and GGG code for glycine. Here, what happened? We have somehow a U has been inserted to a certain coding region. A U has been inserted to certain coding region, like here this. Here it has been inserted. Again I repeat, it is inserted right here between U and A of this UAC codon. So of this UAC codon code for tyrosine now till now. But when a nucleotide uracil is added into this UAC codon between U and A, let us see what changes occur from that point. You can see here now up to this much is okay. Up to this much is okay, fine. AUG code for methionine, CGA code for arginine, UUA code for leucine. But here what happened? Due to the introduction of the U here, the codons get shifted to the next. The remaining codon, the remaining nucleotide of the codon, the remaining nucleotide of the UAC codon has been shifted to the next codon. Like the C which was present right here, the C which is present right here, as addition of the U occurs in this region, so it becomes UUA, and the C codon has been shifted to the next codon because codons are adjacent to each other, and thus it now becomes CGG. Well, you can see due to insertion of UUA now codes for leucine amino acid, and CGG will code for arginine instead of having glycine here. So from that point where the nucleotide is added or deleted within the codon, then what happened? It change in the sequence of the codon. Obviously it will now code for different amino acid from that point towards the downstream. So this is your downstream now up to till now, and this is your upstream. Okay, so from that point the whole codon series of codons will change and code for different amino acid. This is known as frame shift mutation. Well, you can see again here a major thing occurs. You can see right here the UAC codon. You can see the UAC codon that codes for tyrosine. What happened? Somehow this C nucleotide gets deleted, it gets removed. Then what happened? It has to then the first codon of this GGG then actually corresponds with this UAG codon, and thus it becomes UAG now because C has been deleted. To right after C there was G, so this G gets bind with the USG and becomes a stop codon now. So ultimately what happened? The translation process stops prematurely because of just removal of the C from the UAC codon. This type of termination is called, this type of termination leads to your immature polypeptide synthesis. Okay, so this was the first point regarding your genetic code. Let us see the second points. Non-overlapping. Well, the genetic code is non-overlapping. That means the codons do not overlap each other. The codons do not overlap each other like this: AUG, G, C, AUC. Not like this. They do not overlap each other. A non-overlapping code means that the same letter is not used twice for two different codons. So it is saying in simple way that genetic code is non-overlapping. It simply means that the adjacent codons do not overlap, and if overlapping occurs, although it does not occur, if overlapping occurs, then what happened? The same nucleotide of the codon will be used twice for coding of different amino acid. So it is saying that a non, let us see again with different color. So it is saying that a non-overlapping code means that the same letter of the codon is not used for two different codon. But if it was an overlapping, then that same nucleotide can be used for two different codons. But genetic code is non-overlapping. It one letter, one nucleotide will be dedicated for that one codon only. So the nucleotide which was used in previously for a particular codon, that nucleotide will not be used again for the next codon. Like for example AUG, if AUG is coding for methionine, this same A cannot be used for making arginine like CGA. It has to be come from different source. So the nucleotide which was used for previous particular codon, that nucleotide is not used again for the next codon. Okay, so genetic code is always non-overlapping. The codons are located right next to each other. Okay, and the same letter of the codon cannot be used again for different amino acid. That same letter, like AUG is coding for methionine, the same A cannot be used for making arginine. See, it has to take the adenine from different source to make CGA. Okay, so this is what I say about the overlapping and non-overlapping codons. Well, you can see, let us try to explain in an easy way about this overlapping and non-overlapping. Well, you can see right next to each other the codons has been arranged. Also I have given a little big gap here for you to easy to understand, but actually the codons are located right next to each other, but it will make a confusing, so I make a gap here. So you can see AUU. So you can see here AUU. Well, AUU codon codes for a particular amino acid. Okay, very good. Now let us see if it is overlapping, then it must combines with the next codon. Look at this figure. UUG. UUG. So it will code for different amino acid. It will code for different amino acid, right? Then let us see another one. I am giving in now orange. UCU, sorry, UGC, sorry, not UCU. It will be 4-letter code then. It will be 4-letter code then. UGC, UGC. So if it is overlapping sequence, now you can see only 1 2 1 2 3 4 5, only 5 nucleotides are making 3 amino acids. That should not be done. That is, it is saying that codons are not overlapping, actually they are non-overlapping. So overlapping codons will represent in this type of amino acids, which is practically not acceptable. Okay, codons are always non-overlapping. Okay, like for instance, for your simplicity, let us see this two one. CAG coding for one amino acid, CUU coding for different amino acid. Now if it is overlapping, then what happen? If it is overlapping, if it is overlapping, then it will be AGC. It will code for a different amino acid. Then another GCU, okay, coding for another amino acid, right? In this way overlapping sequence are can be said, but codons are non-overlapping. That means each codon is coding for that same amino acid, particular amino acid, and once the nucleotides are incorporated for that particular codon, that particular nucleotide will not be used for the next codon. Okay, so this non-overlapping nature of the genetic code means that the reading frame is read in sets of 3 consecutive nucleotides and that the same nucleotide is not used for the next consecutive codons. Okay, so it directly they are saying it is saying in the genetic code rules that due to the non overlapping nature of the genetic code, it means that the open reading frame is read in sets of 3 consecutive nucleotides. So the open reading frame is always reading in terms of reading consecutive codons. Okay, that is there, it is reading in the way of 3 nucleotide reading. Okay, that makes one codon, and that very nucleotide will not be used for the next consecutive triplets. Okay, just I have said. Okay, next is commaless. Well, commaless means there will be no gap, although I have shown you here gap for your simplicity. I have shown you that how overlapping codons will be look alike, but they are non-overlapping, so that they will code for the specific amino acid, and that nucleotide for that particular codon will not be used for the next codon. Commaless means there will be no space, there will be no intermediary nucleotides between the codons. So there should be no space between the codons. There is no signal to indicate the end of one codon and beginning of the next. So codons are located right next to each other, one by one. So one after another codons are placed. It's not like that there is a stop signal or a initiating signal of a codon. Not like that. Codons are regular, begins with a start codon and ends with a stop codon, and in between there are many codons coding for specific amino acid in order for the translation, for so that they can begin, so that from that codons they will code for the specific amino acid in order to form that polypeptide chain. Okay, next is non-ambiguity. What does non-ambiguity means? Next is non-ambiguity. Okay, so what does non-ambiguity means? Let us see. Wait for a minute. So non-ambiguity, so first let us see what does ambiguous or ambiguity means. Ambiguous means open to more than one interpretation, that is not having one obvious meaning. So non-ambiguous code simply means that there is no ambiguity about a particular codon. Okay, non-ambiguous code simply means there is no ambiguity of a particular codon. And ambiguity means open to more than one interpretation, that means not having one obvious meaning. But codons are non-ambiguous. A particular codon will always code for the same amino acid. So this is the non-ambiguous property of a codon. See, if someone asks what is non-ambiguity of the codon, non-ambiguity of a codon means there is no ambiguity about a particular codon. That simply means a particular codon will always code for the same amino acid. It will not code for the different amino acid. One codon, that is AUG, code for only methionine. One codon, that is your GUG, one codon, that is CUA, will code for leucine only. Okay, will code for leucine only. In that way, well, there are also other codons also that codes for leucine. That means simply means there may be multiple codons coding for the same amino acid, but one codon will code for only one type of amino acid. So that is the non-ambiguous property of the codon. While the same amino acid can be coded by more than one codon, the same codon will not code for two or more different amino acids. This always you remember. One codon will always code for a specific amino acid. There may be more than one amino acid that code, there may be more than one codons that code for the same amino acid, but strictly remember that one codon means one amino acid only. It will not code for different amino acid. For example, in case of codon CUA, so there is a codon called CUA. CUA codon codes for only leucine amino acid. CUA codon codes only for the leucine amino acid and no other amino acid. But there are more than one. But there are more than CUA codons that can code for leucine. Like there is CUA, CUC, CUG, CUU, UUA, UUG. These codons code for, all of these codons code for leucine. All of these codons code for leucine amino acid. But each of these codon will code for leucine only, no other amino acid. Okay, okay. So that is the non-ambiguous role of codons. Codons are always non-ambiguous. But there are some exceptions. There are some exceptions to this rule of non-ambiguity. I have said that one codon will code for a particular amino acid, but there are some exceptions to this rule of non-ambiguity. Well, GUG, as the name suggests, this codon normally codes for valine amino acid, but at the initiating position it codes for methionine. So due to the change in the position of the codon, it can code for different amino acid, as we can see in GUG codon. Well, GUG codon normally codes for what? Valine. But if this GUG codon is placed at the first position of the open reading frame, that means it is placed in the start codon, then GUG will code for what? Methionine. Okay, okay. So this is one exception there is that we can see, but the other codons are have this property of non-ambiguous. One codon coding for one amino acid only, not more than one amino acid. But there may be multiple codons coding for the same amino acid. So one amino acid can be coded by several different codons, and each codon only code for one amino acid only. This is what non-ambiguity talk about. Next is it is universal in nature. Next is it is universal in nature. That means the same genetic code is found valid for all organisms ranging from bacteria to humans. So this genetic code is universally accepted to all living organisms, right? Okay. So the genetic code that is found valid, this genetic code is found valid for all organisms ranging from bacteria to humans. And such universality of this genetic code was actually demonstrated by scientists known as Marshall, Casquet, and Nirenberg in 1967, who found that E. coli, which is a bacterium, Xenopus laevis, which is an amphibian, and guinea pig, which is a mammal, amino acid tRNA use almost the same code. So they have found an experiment, they have conducted experiments, and they have found that about this universality of the code, that these three scientists, Marcel, Casquet, and Nirenberg, in 1967, they have found that E. coli, Xenopus laevis, and guinea pig, their amino acid tRNA uses almost the same genetic code. Well, amino acid tRNA is a step where the tRNA will carry a particular amino acid, and on the basis of anticodon codon pairing, it will be recruited by the ribosomes during protein synthesis. So same type of this genetic code is universally accepted, and they have also stated that genetic code may have developed within bacteria and has changed very little throughout the evolution of living organisms. So the three scientists, Maschel, Casquet, and Nirenberg, have also proposed that this genetic code may have developed within the bacteria first, because they are the actually first organisms that we know that evolved in earth, and this genetic code evolved developed from bacteria, but they have evolved a little modification occur throughout the evolution of the living organisms. Okay, now although this universal nature of codons are true, that all the codons, whether you are observing in bacteria or humans, the codons that codes for specific amino acid is same for bacteria as well as for humans, as well as for mammals. Humans are also mammals, as well as for amphibians, as well as for reptiles, etc., etc. But there are some exceptions. There are some exceptions which has been discovered, which proves that genetic code is not purely universal. How? How can we prove that genetic code is not purely universal? Well, the scientists Nirenberg, Casquet, and Maschel have shown to us that the guinea pigs tRNA, E. coli tRNA, and a Xenopus laevis tRNA carry the same amino acid respect to the coding sequence of the mRNA. But still there are some exceptions, like the mitochondrial DNA. So the mitochondria have their own DNA, and on the basis of evolution, we know that mitochondria was actually a symbiotic bacteria, which have somehow have, due to some causes, it enters into our cells and become associated with the cell, and from that time onward it become a what? It become a part of our cell. So the most notable exceptions are mitochondrial codons, the DNA, the codons which is found in the mitochondria. Mitochondria have mitochondria have their own DNA, which is transcribed and later translated to form protein. Now these mitochondrial genetic codons are little bit different from the universal codons. Let us see what difference is there. Well, if you see the table of the genetic code that has been given in the last of this PDF, you will compare them with the mitochondria genetic code. Well, let us see. UGA. Well, UGA, UGA. This codon is the termination codon. Okay, UGA is the termination codon. Okay, now generally we know that this is the termination codon, but within the mitochondria it will represent something else. Well, the mitochondria which is found in mammals it codes for tryptophan, not stop codon. The mitochondria which is found in Drosophila, which is a fruit fly, also called as fruit fly, the mitochondria of Drosophila, they are UGA will code for tryptophan, same, and the mitochondria which is found in the yeast, they are UGA will code for tryptophan again. Again I repeat for the next codon. Let us see the next codon. AUA. Well, AUA codes for isoleucine normally. AUA codes for isoleucine. AUA codes for isoleucine normally if you see the genetic table, genetic code table. But in mammals, within the mitochondria, of mammals, AUA codes for methionine. Same in the case of Drosophila also. The mitochondrial DNA of Drosophila, they are AUA codon codes for methionine. And in case of yeast, the mitochondria which is found in the yeast, within the mitochondria, there is mitochondrial DNA, and within the mitochondrial DNA, this AUA will code for methionine as well. Well, the CUA is same, not in case of yeast. If you see the CUA codon, generally CUA codes for leucine amino acid, leucine amino acid. CUA codes for leucine amino acid. In mammals also, the mitochondrial, the codons which is found within the mitochondria of mammal, they also code for leucine for CUA codon, and also Drosophila, they also code for leucine, but not yeast. The mitochondrial DNA found in the yeast, the codon CUA within the mitochondrial DNA of yeast codes for threonine amino acid. Okay. AGA and AGG, these codons normally code for arginine if you see in the genetic code table. But in mammals, the mitochondrial DNA in mammals, they are the codons of AGA and AGG codes for termination codon. That means it will stop the translation process of mitochondrial RNA. Whereas the mitochondria of Drosophila, they are AGA and AGG codes for serine, and the mitochondrial DNA of yeast, they are AGA and AGG codes for arginine amino acid. So in this way you can see that how the genetic code is bit different from the mitochondrial codons, from the DNA found within the mitochondria. They have also DNA and they also getting translated to form proteins, but the codons are different from the universally accepted codons. Okay. Okay, so let's move forward now. It's already 1 hour. Now comes the polarity. Now comes the polarity. So now comes the polarity. What does polarity means? It means to say that the genetic code has a polarity. That means the code is always read in a particular direction, from 5 prime to 3 prime direction. We should not read them in opposite direction, otherwise it will code for different amino acid. So it is apparent that if the code is read in opposite direction, that means if we read the genetic code in 3 prime to 5 prime direction, it will specify different proteins, since the codons will have reverse base sequence. See, codons are always read in a fixed direction, that is from 5 prime direction to 3 prime direction, which will code for a particular amino acid. But if the codons are read in the opposite direction, suppose if the codons are read from 3 prime to 5 prime direction, then what will happen? Different amino acid sequence will be formed because the codons are now oriented in the reverse orientation. This is the point, that codons will have a polarity. Next, degeneracy of a codon. More than one codon may specify for the same amino acid. This is called degeneracy of the codon. So there are more than one codons which code for the same amino acid. That is known as degeneracy of the codon. Like, except for tryptophan and methionine. Well, tryptophan and methionine have only one codon coding for tryptophan and methionine. Like methionine is only coded by AUG codon and no more other codons are there. Well, some exception is also there. GUG also code for methionine at the initial stage of translation, and tryptophan is also coded by one amino acid, while the other 18 amino acids have more than one codon. And the codon degeneracy are of two types, partial and complete. So there are partial and complete degeneracy of the codons. Okay, what does partial and complete degeneracy of the codon means? Let us see. So except tryptophan and methionine, all the other 18 amino acids have more than one codon, right? Okay, and the codon degeneracy is basically of two types. One is partial degeneracy, another one is complete degeneracy. Let us see the partial and complete degeneracy. What does it mean? Partial degeneracy occurs when the first two nucleotides are identical but the third nucleotide is degenerate. That means it is changing, but it will code for the same amino acid. Like CUU, the first two nucleotides are constant, but the third nucleotide will be changed. Later on you can see CUC. CUU and CUC, these two codons code for the same amino acid which is called leucine. Leucine. You can see the third letter of this codon is changing, but still it is coding for the same amino acid. That is called partial degeneracy. And complete degeneracy occur when any of the four bases can take up the third position of the codon and still code for the same amino acid. So complete degeneracy means when the any of the four bases takes up the third position. So any of the four bases takes up the third position to code for the same amino acid. That is called as complete degeneracy. You can see here UCU codes for serine, UCC, UCA, UCG. The UC is constant, but the third position is changed by any of the four bases, but still they are coding for the same amino acid. That is called complete degeneracy. So partial degeneracy means the first two nucleotides are constant or identical, but the third nucleotides are degenerating partially. That is called partial degeneracy. Here only CUU and CUC codes for leucine, but there is no CUA or CUG. Okay, that is called partial degeneracy. But in complete degeneracy, when any of the four bases are occupying the third position of the codon, but still they are coding for the same amino acid. Any one of the four bases occupying the third position of the codon, but still they are coding for the same amino acid. That is called complete degeneracy. As we can see in serine, where the third basis is changing, any of the four nucleotides are coming, but still they are coding for the same amino acid which is your serine. Okay, I think you have understood what I have said. So degeneracy At the last, what I can say that degeneracy provides a mechanism of minimizing mutational lethality. So it is minimizing the mutation actually, because most amino acid have more than one codon. A mutant codon may be substituted by another without causing a change in the amino acid. Thus, even if a mutation occurs, the organism still produces a normal protein. So, mutation can occur. It can also lead to frame shift mutation. Suppose if you see the UCU, okay, if this UCU, suppose it is the third position is replaced by A. Suppose mutation occur, but still it will code for the same amino acid, since this UCU, UCE, UCA and UCG, these all codons are complete, showing complete degeneracy, because any of the nucleotides occupying the third base of this codon will code for the same amino acid. So complete degeneracy, and at a certain point of view partial degeneracy, actually minimizing the mutational rate, because if somehow mutation occur, if the third basis is replaced by, or if the third basis is exchanged by some other basis, then there will be no problem somehow. If there is addition or deletion of the base, then there will be leading to frame shift mutation, that we know it will change the whole sequence of the amino acid from that point onwards towards the downstream. But if there is replacement with another nucleotide, then there will be no such structural changes, since due to complete degeneracy, if the third nucleotide of the codon is replaced by any other nucleotide, then it can code for the same amino acid. That is what complete degeneracy is about. So uh, without causing a change in the amino acid, it is getting substituted. So even if a mutation occurs within the organism, it may still produce a normal protein. And another notable feature of this degeneracy is that in most codons, the third nucleotide of the 3' end of the triplet appears to be less important than the first two. Again I repeat what it is saying, that in most codons, third nucleotide at the 3' end of the triplet appears to be of less importance than the first two. For example, alpha alanine has 4 codons, GCA, GCC, GCU, GCG. So the third codon, the third nucleotide of the codon is of less importance. The first two nucleotides of the codon is constant, but the third codon, even if they are changing, the nucleotides are changing, different nitrogen bases are being added, still they are coding for the same amino acid. Another example, threonine. Threonine also has 4 codons, ACA, ACC, ACG and ACU. See, the first two codons are constant. The first two nucleotides are constant. The third nucleotide of the codon is changing. So more importance is given on the first two nucleotides, while the third nucleotides of a codon is of less importance than the first two. So the first two nucleotides are fixed, while the third nucleotide can be filled with any one of the 4 nucleotides. That's the thing that we see in the degeneracy. And that is the reason that why, due to even after mutation of the third position, still it will code for the same amino acid if it shows a complete degeneracy. But if it shows a partial degeneracy like your leucine, which only codes for CCU and CUC, there is no CU, CCA or CCG, it is a partial degeneracy, then what happened, it may affect to some extent. So during protein synthesis, the mRNA codons form base pair with this tRNA anticodon. So as the third nucleotide of the codons have less importance in specifying a particular amino acid, so this will be helpful when that particular codon will be pairing with the particular tRNA molecules. Okay, during protein synthesis, the mRNA codons will form base pair with the tRNA anticodons, and there we see a term called Ubel hypothesis can be brought into consideration. Okay, so this will be discussed on the next lecture. So up to this, I have completed the genetic code. So I will discuss about the Ubel hypothesis in the next lecture. Okay, so that's all for today.